Good evening, ma'am. Good evening, Ajay.
Good evening, sir.
Good evening. How
Good
are
evening,
you?
sir. You can start now.
Yes, ma'am. Thank you. Thank you so much. Good evening, my dear students. So last class, what we have discussed already, you know about what is meant by DNA, what is meant by RNA, what is meant by different types of RNA also already we studied. What are the different type of RNA already studied, my dear? Ajay, how many RNA is there?
Sir, sir, tRNA, sir,
Very good.
mRNA,
Okay.
rRNA,
Okay.
hnRNA, snRNA.
Okay. So among these, which one is very, very essential for the protein biosynthesis or translation process?
Sir, tRNA, sir.
Only tRNA. Is it correct?
Sir.
Okay. Already studied, you know?
Yes, sir.
Yeah. So already you know about the tRNA and rRNA
Sir, rRNA.
and mRNA.
Yes, sir.
These three RNA essential for the synthesis of protein. What are they?
Sir, tRNA, mRNA, and rRNA, sir.
Yes, very good. Because mRNA carry the message from the nucleus to cytoplasm. Okay. And tRNA scattered on the cytoplasm. Ribosome also scattered in the cytoplasm molecules. So which message comes from the nucleus? Okay. So it represents some protein synthesis. Okay. It has some specific codes and codons and amino acids. Corresponding codons joined together with the help of the two components tRNA and rRNA. Ribosome RNA is the factory. Messenger RNA, it's a what is that? What they are going to synthesize? That protein molecules, encoded the protein molecules information. tRNA carry the amino acids from the amino acid pool present in the cytoplasm. So three molecules is very, very essential. Then only the translation process occurs. Otherwise, what happened? No, it will be suffered. Okay. Okay. Already we discussed ribosomal RNA. Already we discussed, isn't it? What are the two types of ribosomes? Already you know about prokaryotic ribosome and eukaryotic ribosomes. 70S, 80S. Subunit, two subunits are each one. Already you know about. In 70S, they divided into two, 50S and 30S. In this category, 80S divided into 60S and 40S. Which one is larger? This one is a large one. Here the two RNA molecules is there. Here the three RNA molecule is there. Already we discussed these things also, isn't it? Today, we are going to discuss about the structure of DNA molecules and structure of tRNA molecules. Okay. See that the structure of tRNA molecules consists of the 5 arms. Already we discussed these things also. What are the arms? One is acceptor arms, T loops, variable loop, and anticodon loop, and D loop. So these are the important arms present in the tRNA, otherwise called as transfer RNA. Already we studied these things also. tRNA was first discovered, Holy et al., 1965, for the yeast alanine tRNAs. He got the Nobel Prize in 1968. Not only that, he chatted with the Nuremberg and Corona. This model is also called as Cloverleaf model. Okay, so already we know about that things also. Okay, so today we are going to discuss deep. So what are the arms is there? 5 arms is there. According to this model, the single polynucleotide chain of tRNAs is folded upon itself to form a 5 arms. A single chain only fold. How many times fold? 5 times. It make it as a arms-like structure. That's why they are called, what is that? Arms. But each one, it has a specific functions. See, the first one is acceptor arms, T-C arms, variable arms, anticodon arms, and DHU, dihydroxyuridine arms. So these are the 5 arms. So the rRNA and tRNA, mRNA essential. The tRNA structure, this is the tRNA structures. There how many arms is there? 5 arms is there. It is single polypeptide chain. This is a single polypeptide chain, but it make it 5 arm structure. Then only the clever leaf model formation takes place. Okay, see that this portion is called as acceptor arms. The amino acids. Okay, acceptor arms is that. Say this one is called as T-C arms. Okay, this one is anticodon loop. This one is called as D arm and loops. So this is the structure of 5'N. This one is called as 3'N. Sometimes they may ask the questions, which side the acceptor arm is present? 3'N. Don't forget, my dear. Okay, see that one by one, clearly what they are doing. What are the types of arms is there, my dear students? How many arms is there? 5 arms.
So 5.
What are they? Acceptor arms, T-C arms. This one is called as extra. Okay, and anticodon, and then D arms. So these are the 5 arms present in it. Particularly tRNA, a single polypeptide chain. It is made up of only single polypeptide chain. They fold 5 arms like structures. Okay, see that one. First one. Types of arms in tRNA. The first arm is called as, what is that? Acceptor arm. The acceptor arms, what they are doing, see that it is include a constant 3' terminal. It is nothing but the tip of 3'. They are having the code CCA is the codon. The sequence. See that one structure, you can understand. CCA. So CCA is the acceptor arm. See, given here. CCA sequence and a 4th nucleotide, either A or G. A or G. Okay, this is the first step. The amino acids molecule attached to the 3' end of the base, the A, adenine, which is known as the amino acid binding site. Don't forget, my dear students. So this is the acceptor arm, means which amino acids is coming in the polypeptide chain. It was received by the acceptor arm. It is located in the 3' terminal. So it is called as amino acid binding sites. So the acceptor arm is also called as amino acid binding sites. Okay, and another one is T-C arms. Here there is a one. It consists of, see here, this is consist of a stem having 5 base pair and a loop of 7 nucleotide. How many nucleotide present in it? Don't forget, my dear students. How many is there? 7 nucleotide is there. It contains, see that the loop contains an unusual base, pseudo-uridine. Okay, don't forget, my dear students. Pseudo-uridine, and hence the name. T-C is also called a pseudo-uridine. What is a loop or base? The loop is involved in binding with the ribosome. Hence, it is sometimes referred to as the ribosomal loop. What is that name? Ribosomal loop. This is called as ribosomal loops. Here you must note it down. 3 point is very, very important. T size is a second arms present in the tRNA. It consists of 7 nucleotides. Don't forget, my dear, 7 nucleotides. Okay, so 7. What is that? Nucleotide is that 7 nucleotides. The loop also, what is that? Unusual base pair. Which one is the unusual base pair? The pseudo-uridine. That's why the symbol given as a size symbol. And it is help for the binding of the ribosomes. So it is called as ribosomal loop. Okay. So third arm is called as variable arms. Variable arms. See, it is in 2 types of one type. There is a loop containing 4 or 5 base and yes, no stem. How many base pair is there? 5 bases. Okay. And has no stems. In the other type, the arms consist of 13 to 21 base and is distinguishable into both stem and loop. This is very, very important. The variable arms. So this is called as the variable arms. See that? This one is called as variable arms. It consists of how many nucleotide is there? See that one. See that one? How many nucleotide? So 13 to 21 bases. Okay. So 3 arms is present in it. And also they are different. And then anticodon arms. See that one? So this is anticodon. Anticodon means which one? The lower part. The lower part is called as anticodon arms. This one joined with the subunit of the, which one you know, ribosomes. I will tell you. Okay. See that one. It is distinguishable into a stem of 5 base pairs. How many base pairs is here? 5 base pairs. And loop of 7 bases. Unpaired nuclei, of which the middle 3 form anticodon. In this category, so 5 base pairs. So, and a loop of 7 bases. What is a loop of 7 bases? I will show you. Okay, see that one. See the structure. So, this is the tRNA structure. So, this is the, which arms is this one? Acceptor arms. Followed by, this one is called as, what is that? T symbol, psi and C arms. There is a small protrude like structure is here in this part and also having the another one in this area. Okay. And the last one is dihydroxyuridine. So this part is called as dihydroxyuridine part. Acceptor arms. Acceptor. Okay. Acceptor arms. This one is called as extra, isn't it? Extra. Okay. So extra arms. Okay. The last one is here, you know. This is called as, what is that? Anticodon. Anticodon. Okay. So this is the structure, tRNA. Here, how many base pair is there? They told 7 base pair here. Started from 1, 2, 3, 4, 5, 6, 7. Okay. Here, whatever it may be. Here, it may be mentioned as here A, G, and C, like this. See, it joined with where, you know, this place is a, see here the ribosome. Ribosome, it consists of 2 subunits, isn't it? What are the 2 subunits? Already you know about. This is the large subunit and followed by the small subunit, isn't it? So here the messenger RNA comes. This one is called as mRNA. Okay, this is the mRNA, isn't it? Okay, so this is a mRNA structure. This one is a mRNA. See here, the structure will be like this. This one, mRNA. The tRNA already, you know, it's present in the cytoplasm. The round shape is there, no? This is called as rRNA. This is called as, what is that? rRNA. So 2RNA already comes, isn't it? The tRNA, where it present, in the cytoplasm, it's present in this place only. See, I will show a different color, then you can easily identify it. Okay. Okay, see that one? No, this one is not sufficient. See that one. tRNA, see here, the structure will be like this. I draw here for your better understand. Okay, so this is the structure. Okay, like this. Another T also here, present in it. Simply I draw here. Okay, structure will be like this. Okay, another also here. Okay, simply I draw this. See here what happened, so the mRNA, it has a code here. Here there is some specific code. Okay, AUG. See, already I mentioned here. Here in this part. This is a part here. Okay, the mRNA corresponding image. That means the mRNA comes here. mRNA, this one mRNA, this one is tRNA. In this portion, I have mentioned in this portion. Okay, see here corresponding A, the join with the which one uracil in this part, uracil and guanine. What happened? Cytosine and guanine. Corresponding bond already in our board. Corresponding bond will be 3. Here also 3. Here 2 bond. So this is the first. This is the formations. So this is the factory. The ribosome, it acts as a factory. Okay. The mRNA is lying on the subunit of both large unit and small subunit. See that structure. It is better for you. See, this is a large subunit. This is a small subunit. In between, what happened? The mRNA platted. mRNA is platted like this. Okay. The mRNA like this. Okay. tRNA already but tRNA in the above units. So this is the way what happened. The protein molecules biosynthesis takes place. That's why the ribosome RNA is a translation factory. Okay. What is that? Translation factory. Okay. See that one? Okay. See. So the 5 base is there and distinguishable into the stem of the 5 bases and a loop of 7 base pair, of which the middle 3 forms a anticodon. Middle 3 form is the anticodon. See that one? You can see the diagram. So here, how many is there? There are so many base pair is there. See here, there is a small line is there within the inside. 1, 2, 3, 4, 5, 6, 7 is there. But 3 is, what is that? In the exposure side. Okay, this is the exposure side that has given here. Okay, see that the 3 middle. So this is the part 3 middle in the form of anticodon. Okay, and the last arms is called as DHU arms. DHU otherwise called as dihydrouridine. So that's why they have given as a name is called as dihydroxyuridine or dihydrouridine. Dihydrouridine. Don't forget, my dear. It is also distinguishable into a stem and a loop. The loop contains an unusual base, dihydrouridine, due to which it is called as dihydrouridine loop or the D loop. This loop is also called amino acyl synthetase binding loop. Don't forget, my dear student, each and every arms, they are having own specific sequences and number of nucleotides as well as different functions also. Okay, so don't forget, my dear, see that one. So this is the structure. This one is called as, what is that? D shaped. This one is called as L shapes. Okay, different types of tRNA molecule is there. This is normally, but the structure is ACC, so acceptor arm sites. This is also acceptor site types and also dihydrouridine anticodon site. But in this category, this one is called as acceptor arms. This is already about TCU arms. This one is extra arms. This one is anticodon arms. This one is dihydrouridine arms. There are 5 arms is there. This is molecular structure. You can see like this. Okay. This is the cleverleaf model. So the same model is also given in the form of molecular aspects. You look like this. Okay. So this is very, very important, my dear students. See that? Sir, in actual 3-dimensional structure, the tRNA is a compact molecule that looks like an inverted L-shaped molecules. Okay. They may ask the questions. The tRNA molecules look like a which shape? Which shape? L shape. Okay. They comes under the heading because it is also one of the protein molecules. So they comes under the headings. Which type of protein molecules? Already you studied. According to their structure, the protein molecules are divided into how many types, my dear? 4 or 5 types. Will you please? Ajay?
Sir.
Yeah. How many types of protein molecule present, my dear? According to the structure.
So 4 types, sir.
Okay. What about the analyst me? Can you? What are the types of protein molecules according to the structure? They may be divided, So I will explain. There are 5 types is the primary structure, secondary structure, tertiary structure, and quaternary structures. Sorry, 4 types, isn't it? Here they told, yes, that's why I'm asking the tertiary structure. So it comes to the conclusion, it is also a protein molecules. Okay, don't forget, my dear students, tRNA, it's look like a L-shaped and also inverted L-shaped molecules. It comes under the heading known as tertiary structure. Okay, what is the next one? It is close to the reality and was proposed for phenylalanine tRNA by Rich and Glick in the year 1974. So this is the structure, phenylalanine structure. Phenylalanine tRNA by Rich and Glick in 1974. So this is also a very, very important, my dear student. This is the structure of which one, tRNA molecules. Okay, don't forget. So this is the another one. It's present in a book. This is the L-shaped. See that structure is look like a inverted L. See, this is the L-shaped structures. This is cleverleaf model. So the tRNA it consists of 2 shapes. One is L-shape, another one is cleverleaf shape. Cleverleaf shape also contain 5 arms. The L-shape is also contain 5 arms. Okay, so that one, amino acid binding site, both are present. Acceptor arm is also there. Okay, anticodon is also present. Okay, other organs is also present. Other arms also present in it. Here they didn't indicate. Okay, because due to yeah.
So could you explain again what the function of anticodon arm is, sir?
Yes, it's anticodon, isn't it? Anticodon is important. See the next one, you can easily identify. See, so this is the structure of TNA molecule. Can you see that, my dear?
Yes, sir.
Okay. The first one, UCA, this is the attached with the tRNA molecule, isn't it? This one.
Yes, sir.
So the opposite side, what they are having, the A, according to the uridine molecules, always combined with the molecules of adenine.
Yes, sir.
Guanine and cytosine. Yes, sir. Same things is also repeated here. So this AGU comes from where, my dear? Where, see that red line. The last word they have given mRNA, isn't it?
Yes, sir.
Okay. This is the already present in the mRNA message from the nucleus.
Yes, sir.
Okay. The corresponding amino acids, this is called as amino acids. Okay. It is spread throughout the cytoplasm. It carried, see, by the tRNA molecule. Corresponding amino acids selected by the tRNA molecule join with this ones. That's why it is called as, this is codon molecule present in the mRNA, exact anticodon carried out by the tRNA molecule. Are you clear now?
So this anticodon arm is basically an arm, sir, which has codon, sir, which is opposite to the codons on the strand,
mRNA.
sir, mRNA strand, sir.
Yeah, correct. Exactly
And
correct.
then so this arm is basically for joining with the codons with on mRNA, sir.
Yeah, very good. Exact joining, and selectable, and also with the bond also it has. Okay, then only that is the
Yes, sir.
reason. Yeah,
So what's
codon.
the bond between them, sir?
Already
Anticodon.
anticodons. What is that? Already told you. Uridine always combined with the molecules of adenine means how many bond is there? Double bond, isn't it?
Yes, sir. So the normal hydrogen bond, sir.
Same
Yes,
as
sir.
it is.
Yes,
Yes,
sir.
it
Yes,
is.
sir. Yes, sir.
Already we discussed, my dear.
Yes, sir.
See, I will show you once again. See, that's why you can easily identify. Okay, see that one. Okay, I will take a new one. So then only it will be better. Okay. So take an example. Already drawn here also already draw the codon also, but here you must note it once how it will be. Okay, so then only it will be better for your understanding. Okay, see that one. See, you are asking anticodon and codon. Okay, this is the mRNA. Okay, just imagine this is mRNA. Okay, mRNA, isn't it, my dear? Okay, it has some codons. Take an example. So many codon is there here. 1, 2, 3, like that. Okay, so many codon is also present in it. So whatever it may be. Okay, it comes from where in the nucleus. Okay.
Yes, sir. Nucleus to nuclear pore. No, sir. It goes.
Yeah. It enter into the nuclear pore until the reach of cytoplasm, isn't it?
Yes, sir. And ribosomes, right, sir? It goes to the ribosome.
Yeah. So it is also very, very essential. I will show you how it will be enter. Okay, see that one. So then only you will understand. So that things also I will discuss. See here, this is considered as a cell. Okay, this is considered as a cell. So inside
Yes,
there
sir.
is a nucleus. Okay, inside there is a
nucleus.
Yeah. So within this one, what happened? There is a, what is this? Chromosomes.
So chromosomes.
Or genes or DNA, isn't it?
Yes,
The DNA
sir.
molecule. Okay. So DNA, what happened? They synthesize the molecule. What is that? mRNA, isn't it? The mRNA. That mRNA is, what is that? Here having. This mRNA is this one. Okay. So this one. Okay. It's carries some message. Here there is a codon is there. Different types of codons already in our board. That things also adenine. So whatever it may be, AUG is a starting codon, and whatever it may be, guanine, cytosine, adenine, okay, and uracil and guanine. So it encoded some specific amino acids and also protein molecules, isn't it, my dear? Okay. This message comes from where, from the nucleus to where? In the cytoplasm. Okay. This is the mRNA. Okay. Isn't it? So this mRNA comes from nucleus. Okay, va?
Yes, sir.
Okay. Then the tRNA and the rRNA, where it present, in the cytoplasm only, isn't it? Cytoplasm only. For example, yes, it's just I put a small T-like structure. So this is tRNA. Okay, small T like this. This is a tRNA, and ribosomal RNA. I put another structures. Then only ribosome I put like this small round only. So these are all ribosome. So how many RNA molecule present in this cell? mRNA comes. This is called as mRNA. Already told you. This one is mRNA. This one is called as rRNA. This one is called as, what is that? tRNA. Okay. Both three is very, very essential. This process occurs within the ribosome. So you can imagine. So this is the ribosome. Imagine this is the ribosome. So large unit, this one is called a small unit. Okay, this is rRNA. The mRNA come and contact with the in between the large unit as well as a small unit in between. Okay, so it carries some message. That's the reason. This region is, what is that? There is a reason. Small subunit, this one is large subunit. Here, what happened? No, the tRNA, see that tRNA present somewhere else. tRNA, see, I give a different colors, then only you can understand. Okay, see that? Okay, this one is not sufficient. Okay, see that. So tRNA present here. Just imagine tRNA is present here in the cytoplasm. Already you know very good. Here the tRNA is there. Okay, it is also present in the cytoplasm. What they will do, you know, exactly. This is the codon anticodon loop. This is the anticodon loop. Anticodon loop. Anticodon loop, isn't it? This plot. Okay. Now what they will do, you know, this anticodon loop exactly match with the AUG. So the AUG molecule is present here in the mRNA. Okay. The tRNA molecules comes from where, you know, in the cytoplasm. Cytoplasm into exactly in this portion. So the position will be like this. Okay. I will put like this small t structure. Okay. So here the sorry. Here the acceptor arm is there, isn't it? This is the, what is the anticodon arm? This is the anticodon. So the anticodon present in the tRNA, the codon present in the mRNA. Okay. But here the joint between the tRNA anticodon loop, what are the molecules they will present? You know, mRNA, what they are having exactly complementary to the tRNA molecules. Here the AUG is present, isn't it? Think about the mRNA AUG. See that? A U G present in the mRNA, isn't it? mRNA, isn't it? mRNA. Here tRNA, what will be happen? Here, what will be the enzyme will be, no, the adenine molecule combined with the molecules of which one, you know? Adenine combined with the molecules uracil only. So the molecule nature, this one. So adenine combined with the molecule of uracil. Uracil combined with the molecules of
adenine, sir.
Adenine. Very good. Here adenine. How many bond is there? Here in this part.
Sir, double bond.
Yes, adenine combined with the molecules of uracil with the double bond only. Uracil combined with the molecules adenine, double bond only. Guanine, how many bond is there?
Sir, triple bond.
Very good.
Cytosine, sir.
Yes, that is the reason. This is called as codon. This is, what is a codon? So exact copy of codon combined with the T anticodon loop side. Anticodon means correspondingly, complementary with each other. That's why it is called as, what is that? This portion is called as, what is that? This part
Sir,
is called
anticodon.
This one is, yeah, very good. Anticodon of the mRNA, isn't it? Now, are you clear now this one?
Yes, sir. So anticodon of tRNA, right, sir?
Yes, anticodon
Yes,
of tRNA. Codon
yes,
is present in the always, whatever it means. Yeah, mRNA
yes,
carry some message and also indicate the particular protein biosynthesis information. Okay. So they are having controlled. You can imagine here. This is a one. They start from 1000. Is 1000. Okay. 1000 nucleotide present in this part. Exactly 3 it will be. So 3 is the indicate the amino acid. This is there 3. So this one is also another 3. So another 3 also. Okay. So what they are codon present in the mRNA, exact anticodon selected by the tRNA molecule from the cytoplasm. So the bond formation also takes place within this area only, this part only. So after that, what happened? You know, after the formation of, what is the exact copy of the molecules, the anticodon, codon united together, these tRNA molecules comes out. See, the tRNA molecules comes out and joint and also carry the another tRNA molecule, and the chain will be continued. You got it, my dear? Are you clear?
So could you explain that last part again, sir? The tRNA.
Yes.
After the bond is formed, sir. What
yes.
are they?
Here I will tell you. Let's see it. There is, and then I will tell you. It's clear. So clear. Then only. Okay. See here. Here. After the bond formation. Okay. So this one, after the bond formation, already you know the AUG exactly match with the double bond double bond,
Yes, sir. Triple
double bond. Yes, a triple bond. Sorry, yeah. Okay.
Yes, sir.
Yes, see here. So I will explain this. Here the C is the, so here the
Okay.
A is the,
Yes, sir.
yeah, here also U.
You, sir.
After formation, it takes place, isn't it, in this bond?
Yes,
Okay.
sir.
Okay. Here what happened, the tRNA carries, isn't it? tRNA. So this is the tRNA.
Yes, sir.
Yes. Okay, I will tell you. Okay. So this one, the tRNA molecule. Exactly the tRNA molecule here. After the formation, what happened? The tRNA molecule comes out from the ribosome. It's a comes from the
Yes, sir. Ribosome, sir.
ribosomes. It comes from. There are some dissociation from the chain. That is very, very important. They need some, what is that? Composition also. They need some physiological changes occurs within the ribosomes. See that one?
Yes, sir.
So it
Sir,
comes
all
out.
this have-
Why? What one?
Sir, so all this happens inside the ribosome. So after
Yeah,
it has
yeah.
bonded, it comes out of the ribosome.
Yeah, yeah. After bonded only it comes out.
Yes, sir.
Okay. Then see that this is the tRNA, isn't it? This tRNA after it comes out. Okay,
Yes,
the
sir.
chain will be continue like this. The chain will be continue like this. Here, the chain will be going on properly. It's a double standard and codon and anticodon. Okay.
Yes,
The
sir.
tRNA again re-enter to the another components. For example, suppose if it is the mRNA contained another codon like this, capital C, capital
Yes.
C, capital C, like that, the tRNA molecules carry the another molecules. How it will be? Will you please?
Sir, it will be GGG, sir.
Correct,
Triple bond.
exactly correct. So the molecules will be like this. Okay, so just I put a small diagram only. Okay, just imagine. So here G and G and G is also coming next. Okay, likewise there are so many. Yeah.
Sir,
Yeah.
this will be the same tRNA as before, sir, or different one, sir?
No. There are number of tRNA. It may be lining on it.
Yes,
Here,
sir.
suppose if you take an example, this one is 1.
Yes,
This
sir.
is a 2, 3,
Yes, sir.
4, up to 100.
Yes,
Whatever. How many nucleotide is there? We do not know. Okay. How
Yes, sir.
many nucleotide?
Sir,
There are so many.
Sir,
Yeah.
so after it, sir, so it bonds and then that codon will remain together on that stand, and then this tRNA rest alone goes out of the ribosome, sir.
Yeah, correct. Exactly. It comes
Yes,
out after the formation
sir.
of the bond. What happened? It dissociates from the chain.
Ribosome, sir.
Ribosome.
Sir,
Yeah,
what
yeah.
happens to it, sir, then? That whatever is left behind, sir.
After that. So
Yes,
until
sir.
until the stop codon.
Yes, sir. So
Okay.
until the stop codon, this process happens, sir. But what will happen to the, sir, like the tRNA gives the anticodons to the codons and then it exits the ribosome, no, sir?
Yeah. Because why, because the codon and anticodon united means it's encoded with some amino acid and also the protein molecule. Suppose we need, take an example, sir, Ajay. So you need some protein molecule. Okay, for example, hair or nail. Okay, hair is a keratin protein, and
Yes,
also
sir.
nail is a keratin protein.
Yes,
But
sir.
both are different because hair color is black and nail color is white in color. Okay,
Yes, sir.
so they are having different amino acids, isn't it? Amino acid sequences. Then only the structure will be, and also color will be changed.
Yes, sir.
Okay, here what happened, so this is considered as a protein molecules of keratin means keratin. Just imagine
Yes, sir.
keratin.
Yes, sir.
Okay, so it has a specific amino acid sequences in this part. The particular amino acids codon started from the AUG and finished with the stop codon. When the
Yes, sir.
stop codon comes here, automatically the keratin protein biosynthesis will be stopped.
Yes, sir.
Okay. Next, I need which one, globulin protein or immunoglobulin
Yes, sir.
or heparin protein. Mean it started from the new aspect.
Yes, sir.
It is also started from the where the another one means another. So same, not only the single ribosomes. How many ribosomes present in the cytoplasm? Number of cytoplasm. Yeah,
Yes,
polysomes.
Lots,
They are
sir.
they are called as polysome. For example, take an example. I have drawn here only one ribosome here, isn't it?
Yes, sir.
But what happened in this part, there are so many ribosomes there. See that one? So like this. Okay. So the so many ribosomes like this. This is called as polysomes. Okay. We will discuss how it will be. All those things we will discuss. Now we are studying which one, what are the types of RNA, tRNA, mRNA, and rRNA. Now what we are discussing, tRNA, what are the arms is present, what are the, what is that? Functions they are doing. That things we are studying. Okay, ma?
Yes, sir.
Okay. Okay, my dear. Okay. See that one? So it is easily identified. See, this is the structure. So you can understand this is the tRNA
Sir.
molecule. Yeah. Yes, my dear. Go ahead.
Sir, what will happen to that structure, sir, after the anticodons and codons?
There is no any change in the structure, my dear. There is no any
Sir.
changes.
Sir, what will happen to it, sir? Sir, like it's leaving the ribosome and going, sir. Ribosome is undergoing physical change, sir. What will happen to the leftover this structure alone, sir?
Structure, there is no change. After the finishing of it's also going on continuously. There is no any change in the tRNA, mRNA, and rRNA. But particularly mRNA it will be changed. Because codon it only present in it, isn't it, my dear?
Yes, sir.
So when the tRNA and rRNA come in contact close together, as a result, what happens? The protein molecule biosynthesis takes place.
Yes, sir.
Okay.
Sir, so we move on to the next one, sir.
Yeah. That's all. Continuously
yes,
it's going
it will
on.
enter a new ribosome, sir.
No. It will not produce the new ribosomes. Only the ribosome present as it is. It is nothing but the factory. Factory is moving or not?
Yes, sir.
Factory is any changes it's occur?
No, sir.
Because present as it is. But what they are doing, the biosynthesis takes place within the ribosome only.
Yes, sir.
So what they are doing, codon and anticodon united with the, they are doing the work, what they both codon and anticodon bonding function or selection process only occurs within the factory.
Yes, sir.
Okay, see that one. See that one. So this is the structure of this one is called a serine. See the 3 diazymes and also 5 diazymes. This is the structure of the tRNA molecule. Here the anticodon is the corresponding codon. So you have to imagine mRNA, what they are having, exact anticodon was selected by the tRNA molecule. After the formation, what happened? No, it can move along the, which one move along the mRNA molecules, move along. So here in this part. After that, the tRNA comes out. After that, it comes out and also it's also collect another amino acids. So here the serine is there. Here the tyrosine is there. Okay, this is another codon. Likewise, how many codons anticodon present in it? How many codons are present in the mRNA? Exactly combine the complemented to the tRNA molecules also. Okay. Each and every cytoplasm it contains number of tRNA molecules. Each one it carries a specific amino acid. Which one is suitable for the codon? It can be easily joined within the ribosome only. Okay. After the formation of what is that? Codon and anticodon, with the help of the bond, hydrogen bond, what happened? No, the tRNA dissociated from the ribosomal RNA and also it's also enter into the cytoplasm and carry the another molecules. Okay, so see that one. The functions of tRNA, the acceptor arm binding site of the amino acids, DHU arms binding site of the amino acyl tRNA synthetase enzyme. Don't forget, my dear, this is very, very dihydroxyuridine arm binding site of the amino acyl tRNA synthetase enzymes. Anticodon arms already binding site of mRNA subunit, messenger RNA subunit. These size C arms binding site of the ribosomal small subunit. So these are all called as what is that? tRNA functions.
So,
okay. Yeah.
So this amino acyl tRNA synthetase enzyme, sir.
Yeah.
What is it used for, sir?
So it is used for the binding site, means what is that? The amino acids want to bind, then only. What is that? This one. So this one, what is that? AGU.
Yes, sir.
So what is that meaning?
Codon, sir. Codon.
It indicates some amino acid,
Yes, sir.
Amino acid can bind anywhere. It's possible.
Yes, sir.
It is not possible.
Sir, no, sir. It is not possible, sir. Sir,
yes.
It can finally bind on that acceptor arm, sir.
Just Ajay, you should know the enzyme substrate complex, you know, enzyme substrate, do you know that one? Exact copy of enzyme, they can bind with the exact copy of substrate only, isn't it?
Yes, sir. Yes, sir.
Likewise, what happened here, the codon so bind with the exact copy of the anticodon.
Yes, sir.
That was initiated by some of the chemicals, substances, or enzyme. That enzyme is called as, what is that, you know? The amino acid
tRNA synthesis.
that is
Yes, sir.
enzyme.
Sir, it's
Okay.
basically used for binding, sir, of the
Yeah.
mRNA subunit, mRNA
Yeah.
codons to these anticodons.
Yeah. So see here, we
Yes, sir.
all are doing the same process only. Here, the binding site of the amino acids, first point, acceptor arm. That is the reason here. You see that this part is acceptor arm, isn't it? Here, the amino acid binding site.
Yes, sir.
Okay, because which protein molecule they need, that things here. So it carries here. This is also amino acid. This is also amino acid. Here, there all are so many amino acids. Here only GGEC also mentioned. Okay, when it comes, for example, this part comes, you know, this part comes here, you know, imagine the ACC, you know, this part when comes here, it can move in this part, my dear students. See here, like this, likewise it moves. Do you understand, my dear?
Yes, sir.
This is the amino acid, isn't it? Amino acid attachment. Okay.
Yes, sir.
It can move like this. It can move like this in this part. When it reaches the anticodon arms, okay, anticodon arms, that time what happened? No, the tRNA having alanine. See here, GGC they have given as a alanine. Okay.
Yes, sir.
The mRNA corresponding combinations, CCG it carry.
Yes,
Suppose likewise it joined together, then only it will be the protein biosynthesis takes place. Okay, see that this is very, very important. See here once again, these molecules move here likewise. Okay, UCA. When it AGU present in the mRNA, exact copy of the UCA comes from where, you know, in the acceptor arms, in this area only. Okay.
Yes, sir.
Likewise, what happened? The molecules all synthesized and also released from this area completely. They are all occur in the ribosome only. Okay. I will tell you that things also I will tell you, because not yet enter into the translation process. Only we are studying the tRNA structure. What is the main functions of the arms only we are studying. Okay, dear.
Yes, sir.
Okay, see here. So next one is mRNA. First we studied ribosomal RNA. Second we studied tRNA. Third we are studying, which one, you know, mRNA molecule. What is meant by mRNA? mRNA nothing but messenger RNA. What messenger? The particular type of protein you should synthesize. That message comes from where, you know, the nucleus of the cell. So that is why it's called as messenger. Okay, messenger RNA. What is the meaning of messenger RNA? Once again, repeat for you. The particular amino acids, a particular protein molecules synthesizing message present in the RNA. That's why it is called as mRNA. This was first proposed by, the name of the scientist is called as Jacob and Monod. In the year 1961, proposed the name of messenger RNA for the RNA carrying information for protein synthesis. Already I told you. Okay, last slide, I draw it as a cell structure. Inside there is a nucleus. Nucleus, they need some protein molecule for their growth or metabolism or physiological process, whatever it may be. It can be transferred through the mRNA, because mRNA carry the information from the nucleus to cytoplasm. After enter the cytoplasm, what happened? You know, the ribosomal RNA as well as the tRNA help together. As a result, the protein biosynthesis takes place. After the formation, the polypeptide formation takes place. Okay, when the stop codon comes, at the time, what happened? Automatically, the translation process completely stopped. Okay, this is the process. Okay, and another mRNA from particularly taken example, heparin protein biosynthesis. Okay, that message also carried out by the another mRNA. The same process also repeated for the synthesis of heparin protein. Suppose taken examples of another protein molecule, immunoglobulin or globulin, any other protease enzymes, taken examples, they can carry some message from the cell, and also it's also enter into the cytoplasm. Again, the cytoplasm, they are having rRNA and tRNA through this, with the help of these two molecules. Again, they synthesize within the different types of protein molecules, polypeptide chain. Okay, my dear. So yeah, the mRNA, messenger RNA, Jacob and Mona 1961 proposed the name of messenger RNA for the RNA carrying information for protein synthesis from the DNA genes to the site of protein information formation, ribosomes. It is a single standard straight chain, every short-lived and largest, and it constitutes about 3 to 5% of the total cellular RNA. Already we discussed it. mRNA, how many percentage present in it? 3 to 5%. What about the tRNA? 15% present. What about the rRNA? 80% present. Okay, see here. The molecular weight of an mRNA molecules varies from 25 into 10 to the power of 3 to 1 into 10 to the power of 6. This is the molecular weight of mRNA. So these are the 3 RNA molecules, very, very essential. See the structures, types, mRNA, tRNA, rRNA. Sedimentation coefficients 65 to 255. Minus 15, here 55. The molecular weight, my dear student, this is very, very important. Molecular weight in Dalton, I have given the mRNA 25,000 to 1 lakh. The tRNA 23,000 to 30,000. The rRNA minus 35,000 Daltons. What about the number of nucleotides? See here, 75 to 3,000, 75 to 19, minus 100. Percent of total cell RNA minus to 16. So these are all rRNA subunit, 5S, 16S, 23S. These are all rRNA subunit. So they are all comes under the heading 82 total cells. This is the another major class of ribonucleic acid in E. coli. They have given in the prokaryotic manner. What are the prokaryotic contains? What are the RNAs? Sedimentation coefficient how much? Molecular weight is how much? And number of nucleotide residues present in the E. coli molecule, and percent of the total cell RNA molecules also given. Okay, see that one. So this is the structure I already told you. See here, how the mRNA molecule comes from. Say this is the structure of nucleus. See that nucleus is here, outside. This is the inside. This is the nuclear pore. The mRNA molecule synthesis takes place with the help of, what is that? A transcription takes place. This is the DNA molecule. Produce the single standard RNA molecule. After the formation of single standard RNA molecules, it carries the message. That's why it is called as messenger RNA. It comes from the nucleus to cytoplasm through the way of nuclear pore. Are you clear, my dear? Ajay? Are you clear now?
Yes, sir.
Okay. This is the nucleus, isn't it? DNA molecule present inside. The RNA molecule synthesis takes place. That is called as transcription. After the formation of single standard RNA molecule, carry the amino acid sequences and also carries the message. That's why it is called as messenger RNA. After the completion of the mRNA molecules, it enter into the nucleus to cytoplasm through the way of, which one you know? Nucleopore. Nucleopore is nothing but the outermost layer. There is a small space in between the two adjacent layer. It's called as nucleopore. After it enter the cytoplasm, it collects the ribosomes and tRNA. The protein biosynthesis takes place. Okay, see that one. It is also called as informational RNA or template RNA. The lifespan of mRNA is 2 minutes in prokaryote and few hours in eukaryote. Don't forget, my dear. This is very, very important. This is very, very important. Okay. The lifespan of mRNA is 2 minutes in prokaryote and few hours in eukaryote. Its sedimentation coefficient is 88S Swadberg unit. Since mRNA is transcribed on DNA, its base sequence is complemented to the segment or DNA on which it is transcribed. DNA to RNA, simple. Introns, very, very important, my dear. What is my introns? Non-coding sequences are called as intron. Mostly the non-coding sequences or introns are absent in prokaryote mRNA and present in eukaryote mRNA. Don't forget, my dear, this is very, very important. Okay, the introns, what is the meaning of intron? In between the two sequences, take an example. So this is called as, okay, take an example. So I will tell you. So this is the polypeptide chain, imagine. Okay, so here what happened? There is a exon, likewise, imagine these are all called exon. What is the meaning of exon? This is a small fragment like structure present in the nucleus but encoding the amino acid. What about in between this part? So this part is, what is that, see that, this part. Okay, and so these part are called as intron. Intron. Okay, intron. How the mRNA formation takes place? mRNA, listen here, my dear, this is very, very important. mRNA, listen here, the complete introns are, what is that, cut. So as a result, what happened? The exons are united together. As a result, the formation of, what is that? See that one? This one, and this part, and this part. Completely united as a result, the formation of the mRNA. So this is the first part, this is the second part, this is the third part. Okay, like that, imagine. So this is the first one. See that one? 1 and 2 and 3, and also this one is 4. Okay, it encoded the codons. But in this case, introns, they will not encode or non-coding substances here. It completely cut this portion. Completely, they cut this portion, non-coding portion, completely cut. As a result, you will get the red line only. So red line only, you will get it. This is called as, what is that? mRNA. Are you clear, my dear, in this part? A particular nucleotide, it contains exon and intron. Exon is a coding sequences. Intron is a non-coding sequences present in the two exons. Okay, the exons are removed and also joined together. The introns are completely removed. So that is also very, very important. The introns are removed, you will get the extrons. The extrons are united together and also carry the message. That's why the mRNA formation takes place. This is the formation. Okay. Okay. See that one. Okay. See that one. The sedimation coefficient already told you. Introns, non-coding sequences are absent in the prokaryote. Don't forget, my dear, mRNA and present in the eukaryote. We draw the slide, that is called as eukaryotic RNA molecules. That because it has two molecules, intron and extron also present in it. But at the same time, in prokaryote, what happened? There is no any, what is that? Molecules. Any molecules, what is that? Intron are absent. That means extra only present in it. But in the case of eukaryote, extron and intron also present in it. That is very, very important. Okay, don't forget. See that one. It is also called as informational rRNA. Already we discussed these things. See that it is made up of approximately 900 to 1,500 nucleotides. How many nucleotides is that? 1,500 nucleotides. It is a monocystronic in eukaryote and polycystronic in bacteria and phages. Okay, so this is the monocystronic. Monocystronic, single molecule, same type. Polycystronic means more than one. Okay, in bacteria and phages, several and termination sites also present in it. There is no processing in prokaryote. Once again, repeat for you. That mRNA molecules is made up of which one 900 to 1,500 nucleotide is present in it. At the same time, monocystronic in eukaryote and polycystronic in bacteria as well as the phages, lower animals. There is no processing in prokaryote. It is also another one. See that one? So mRNA is there. This is a starting codon. This is a stop codon. See here, unrelated region. This is called as 5' is also having unrelated region. This one is also unrelated regions. Okay. So here the stop codon is there. This portion only the protein coding region. This area, it may be considered as a 1 to 1000. Adenine, guanine, cytosine, like that. In this two side, what happened, there is a non-coding region present in it. It's simply called as untranslated region, UTR. Okay, this is called as UTR region. See the next one, you will get it idea. See, this is a UTR region. This is also UTR region. This is a protein codon regions. Okay, this is poly A tail. So this is another thing. The entire chain is there, but in between what happened, there is a UTR region, untranslated region is also present in it. At the same time, here the protein coding region also segment is also present in it. Okay, this is the structure of the eukaryotic mRNA structures. See the structure, you can easily identify. See these two are unrelated and transform regions. So this is also untransferred. Protein 1, protein 2, protein 3. Eukaryotic mRNA and monocistronic. Okay, so just remove it all the part. Which one The untranslated region here. The protein is in 1. So this one is here and also unrelated. So this is the structure. So the eukaryote, this one is called as prokaryote. This is called as prokaryote means what is that? Polycistronic. This is monocistronic. You know the difference between polycistronic and monocistronic. Okay. More than one codon present in it. That is called as cistron is nothing but the small segment present in the mRNA. It encoded the protein molecules. Suppose if it is present more than one cistron in the mRNA molecule, it is called as polycistron. Suppose if it is present in the only one, it is called as monocistron. The polycistron mostly present in the prokaryote, otherwise called as bacteriophage or bacteria in lower organisms. And monocistronic mostly present in the higher organisms such as eukaryotes. Okay. But in this category, multiple translation start sites present in multiple translation start sites. This is called a starting sites. So here only the one translation site is present. That's all. Okay. So it is opposite to the eukaryote. Highly developed organism is here. What happened now? Only one is present, one starting code, one starting site is present. At the same time, monocistronic in nature in higher animals. Lower polycystonic, more than one translation start sites also present in it. That is the difference between. See that one? Okay. So the same things also given here. Okay. These are the, what is that? 1, 2, 3. So this is called as prokaryote. See here, the only one is there. So this is the only one. So here, there is no codon. So this is the monocistronic and polycystonic differences and also examples. Okay. This is very, very important, my students. So many times they were asked the question in this part. Okay. So remaining things, I think we will finish it up to this level. Remaining things we will see the next classes, my dear students. Today, what we discussed, we will summarize once again. So last class, what we have discussed, what is that, ribosomal RNA and tRNA. So now we discuss in this part, what are the arms present in the tRNA molecules. There are 5 types of arms also present in it. What are the arms? Acceptor arms, T-side arms, variable arms, and anticodon arms, and DHU arms. Okay. So this is the structure of the tRNA molecules, otherwise called as clever leaf model, 2D models, all those things. What are the arms also present in it? That things already we discussed. Okay. Each one, it has a specific functions also. Okay. And then see that the structure of the clever leaf model was first discovered. How it will be look like a structure, L-shaped molecules. Okay. It is also called as tertiary structures. Tertiary structure. It is closely to reality and was proposed by the phenylalanine tRNA by Rich and also Gluck in 1974. Already the things also already we discussed. This is the structure of the which one molecules, tRNA molecules, and also 3D structures. Okay, and L-shaped, okay, structures. And also, and the codon anticodon regions, how it will be joined with the codon molecules of the mRNA with the anticodon of the tRNA molecules. So functions of the tRNA molecules is the acceptor arm, DHU arms, and also anticodon arms and C-Cy arms, how they will be bind with the another molecules, amino acids binding sites, how it will be. And next one is the mRNA. mRNA was first discovered by Jacob and Monod. And what will be the nature, where, when, how? 1961, they discovered the molecules, and how many molecules, how many percentage present in the cellular aspect, mean 3 to 5% already we discussed. Okay, the molecular weight will be 25 into 10.3 to 1 into 10 to the power of 6 Daltons. Okay, that things also we'll discuss. How it will be produced, transcription takes place within the nucleus molecule, how it will be enter in from the nucleus to cytoplasm through the way of which one, nucleopore, that things also we discussed. And the lifespan will be 2 minutes, one be in prokaryote and few hours in the eukaryote. Which one? mRNA, don't forget, my dear. Okay, it is also called as what is a template RNA, because it is a small segment only. mRNA is nothing but a small codon, a small polypeptides only. The sedimentation coefficient is very, very important of the mRNA, 8S. And also introns, non-coding sequences are absent in prokaryote, and mRNA is present in eukaryote. This is also very, very important things. How many nucleotide approximately present in 900 to 1500 nucleotide present in it, and also it exists in two forms, monocystonic and polycystonic. Monocystonic means mostly present in the eukaryotes. Okay, polycystonic means which one, and bacteria and bacteriophage. What will be the structures? Already we saw it. See that one? This is the structure of the monocystonic and the polycystonic. And how many starting sites also present in translation sites here? The more than one polycystonic or acting sites, starting site present in it, it is called as prokaryote. Suppose if it is present in only one, is called as what is that? Eukaryote. Okay. Thank you for the participation, my dear students. We will see the remaining things on the next classes. I think 3 or 4 slides only present in it. Okay. So we will see the remaining things on the next class. Thank you, my dear. Thank you so much.
Thank you, sir.
Thank you, ma'am. Thank you, ma'am. Thank you.
Thank you, sir. Thank you, sir.
Dhanarishmi also. Okay. Thank you, ma'am. Thank you.